Hello everyone, welcome to NEET YUG. Here I am going to explain Crescelation acid metabolism, a very important pathway for your competitive examination. Okay, where exactly it is seen? This pathway is seen. Normally you see them in Crescelation family plants like Apuntia, Bryophyllum, Agave and even the pineapple also. Here what is important is they exhibit scotoactive stomata. That means this stomata opens only in the night time, closes in the daytime. The reason is to avoid the loss of water because of transpiration. Here let me just write how exactly this pathway goes. Almost similar to your C4 pathway, but the reactions like synthesis of organic acids takes place in the night time. That is very important in this CAM metabolism, that is organic acid synthesis normally takes place in the night time. So in the night time you see them, hence because of the synthesis of the organic acid the stomata opens in the night time, and in the daytime what's going to happen is the same organic acids which were synthesized in the night time get converted into some other compound which now synthesizes the hexose sugar, hence the stomata now closes. It is very important to understand. Now where exactly this CAM takes place in this Crescelation plants? First I have drawn a box here. This box is the guard cell. Okay, so that is understood that guard cells also take place or take part in this particular CAM metabolism. And what I am going to do is I am going to draw a big vacuole here. The guard cells contain a big vacuole. They also play an important role in opening and closing of the stomata. Here I am going to draw one box. This box is very close, that is in close association with the guard cell. This box is compared to a cell that is nothing but the mesophyll cell. Okay, mesophyll cell. What is interesting in the mesophyll cell of these plants, that is CAM plants or desert plants, is they exhibit both the types of enzymes, that is they have rubisco and also pep carboxylase enzyme. So they have both the types of enzymes with them. Because of the presence of both the enzymes, the rate of photosynthesis will be very low compared to C3 plants and C4 plants. Out of these three types of plants, C4 plants exhibit, that is has higher rate of photosynthesis. Next is C3. Next, last one is CAM plants, Crescelation family plants. Okay. Now, how exactly this pathway is initiated? To understand that, let me just divide into two portions. This portion is the reaction that is occurring in the night time. So let me write here during night time, and in this case the reaction is occurring in the daytime. Okay, this portion is daytime, so it is also applicable to guard cells, mesophyll cell, guard cell. Okay, so what we do is we write the reactions. First is the phosphoenol pyruvate, which is the primary carbon dioxide acceptor in the case of CAM, similar to your C4 pathway. And because it has got one enzyme, that is pep case or pep carboxylase enzyme. Okay, carboxylase enzyme, which now produces oxaloacetic acid, which is a 4-carbon compound acid, organic acid. This oxaloacetic acid now gets converted into malic acid. We get malic acid. For this we need one enzyme, that is malate dehydrogenase, similar to your C4 pathway, Hatch and Slack pathway. And what is required is NADPH. The assimilatory power is required for the conversion of OAA into malic acid. We just write it. Okay. What is the fate of this malic acid, which is now synthesized in the night time within the mesophyll cell? Now this malic acid is now transported into guard cell and then enter into the vacuole of the guard cell. In the guard cell vacuole, they get converted into, the malic acid itself get converted into potassium malate. Hope you can see this potassium malate, that is in the night time. The potassium malate now forces, or it is responsible for the opening of the stomata. So potassium malate now allows the guard cells to move so that the stomata opens. That is understood that stomata in the Crescelation family plants opens in the night time. Why? The reason is the malic acid. Okay, because it gets converted into potassium malate. Now, in the daytime, the same potassium malate is now converted back into malic acid within the vacuole of the guard cell. Again, it is transported back into the mesophyll cell. In the mesophyll cell, the malic acid breaks to produce carbon dioxide and pyruvic acid. We write here pyruvic acid. What is the fate of this carbon dioxide? The carbon dioxide now enters C3 pathway. So now it is understood that C3 pathway in the sense hexose sugar is synthesized. Okay, sugar is now synthesized. Now this sugar gets converted into starch. Starch, normally the stored food of the plant. Okay, this starch can be used whenever plant requires energy, or the same starch can get converted into some other product. Let us see that this pyruvic acid now gets converted. Okay, I just would like to give you one more information that the malic acid is there, right? The same, that is NADP malic enzyme is required for the conversion of malic acid into pyruvic acid. Okay, here what's going to happen? NADP now reduced to NADPH. So we got the lost NADPH here, from here, see here. So we got it back, NADPH. Okay, so this pyruvic acid now transforms or get converted into glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate. Okay. Next, the same glyceraldehyde 3-phosphate now enters glycolysis pathway, that is in the night time. All these things are happening in the daytime, that is still glyceraldehyde 3-phosphate, that is malic acid till glyceraldehyde 3-phosphate. It is understood that sugar is synthesized in the daytime when the stomata is closed. Here, the glyceraldehyde 3-phosphate that is enters into glycolysis pathway will now form phosphoenol pyruvate. Phosphoenol pyruvate is now formed, and again the phosphoenol pyruvate now synthesizes phosphoenol pyruvic acid, that is because of the enzyme called as PEP synthetase. Okay, hope you can say this. And this PEP is now ready for another fixation of the carbon dioxide, and the process repeats. This is all about crescelation acid metabolism. Got it? Why the rate of photosynthesis is very low compared to C3 or C4 pathway is it has both the enzymes. One is PEP carboxylase and rubisco. So the rate of photosynthesis is low in crescelation family plants. Hope you understood this session. Thank you.